So we'll continue discussing the anatomy of flowering plants. So now we will discuss the ground tissue system, basically organized into two different layers, external and internal. The external consists of endodermis cells and cortex cells, whereas the internal consists of stele and pith. So endodermis, which is made up of parenchyma cells, innermost layer of cortex. There is no intercellular space and it has a waxy deposition in the form of radial and transverse band is called Casparian strip. So this is how the Casparian strip is present in the endodermis. Okay. Then cortex cells, which are maybe parenchyma, collenchyma, and sclerenchyma, and they consist of all these in different way, and they consist of few layer of cells for protection and store of water, which are known as hypodermis. We'll see where hypodermis is present, how these different cells are organized in the root and stem uh, in this video. Then internal stele, which is made up of parenchyma, and it also consists of complex tissue like xylem and phloem, and consists of tissue of inner endodermis. Pith, which is again made up of parenchyma and is the innermost ground tissue that stores starch, fatty substance, tannin. So if you look at the pith, how it is organized in monocot stem and dicot stem and root. So monocot root, they have large pith. Even dicot stem, they have large and well developed pith. But if you see dicot root, the pith is very small or absent, but you cannot differentiate the pith in monocot stem. Now, pericycle is you can say different types of cells which are surrounds the vascular bundle and are present between the endodermis and vascular bundle and consist of parenchyma, sclerenchyma cells, and it is the seat for the origin of literal roots. Plays significant role in the secondary thickening of root, and the core cambium arise from here. So we will see all these how it takes place. So if you look at the diagram, transverse action of plant root or shoot, this is how you will see epidermal cells, they are outside, and then you will see the ground tissues and the vascular. So three different tissue system is clearly visible here. Again, the ground tissue are divided as the external and internal. So here again in the LS also you can see internal and external clearly, vascular tissues, and then you can see the epidermis. So now next is we will see the vascular tissues which consist of xylem and phloem. So here we will see the arrangement, how they are arranged. We have already seen that they consist of what all element or what all cells in a tissue, complex tissue. So types of vascular bundles on the basis of how they are arranged, they are divided into three categories: radial, conjoint, and concentric. Radial, here the xylem and phloem, they are in the alternate patches on different radii, usually occurs in the so you can see xylem and phloem are in different bundles. In the conjoint, the xylem and phloem are in the same bundle, in which xylem is toward interior and phloem is toward exterior. Now, depending on their arrangement, they are divided into two categories: conjoint collateral and conjoint bicollateral. So, collateral consists of xylem and phloem, where they occur in the same radius, xylem in the inner radius and phloem in the outer. So, you can see here xylem toward inside and phloem toward outside. Now, again, the collateral are said to be open when cambium is present, and they are said to be closed when cambium is not present. So usually you will see in dicot stem the open one. Usually dicot have the cambium, whereas monocot they don't have cambium. But collateral and conjoint you will only see in the stem, not in the root. Okay. Bicollateral are the open vascular bundle consists of xylem in the center and which is surrounded by phloem on either side. So it consists of two cambium, two phloem, one xylem on the same radii. You can see like this. So xylem in the center, phloem on the either side, and then phloem is separated by the cambium. Sorry, phloem is separated from xylem by cambium. Now concentric, the xylem and phloem are in concentric circles. Like here, the xylem is surrounded by phloem. This is called amphicribal, where here the phloem surrounded by xylem. Okay, so this is amphivessel. So these are different arrangement which you need to understand. Now we'll just see basic anatomical difference if you study the transverse section of root and stem. So what is the difference between root and stem as far as these different cells and their arrangement is concerned? So cuticle, which is the outermost layer to the epidermis, is absent in root but it is present in shoot. The root hairs are unicellular, whereas if you see the epidermal hair of shoot, they are multicellular. Endodermis is well defined in root. In shoot, it is absent. Roots vascular bundles are radial, that is alternate, whereas in shoot they are conjoint and collateral. Um, Xylem is as exarch in root, whereas in shoot it is endarch. Now, if you see the difference between the monocot and dicot root, here the epidermis is present throughout, whereas in dicot the epidermis is replaced by periderm. Monocot number of vascular bundle are more as compared to dicot. In the dicot it is 2 to 6, and pith is large in monocot as compared to dicot, and cambium is always present in dicot and is always absent in monocot. Now, if you see the monocot and dicot stem, endodermis is absent here, whereas in dicot stem it is present. Pericycles surround each vascular bundle in monocot, whereas in dicot the pericycle form the cap to the vascular bundle. Pith is not differentiated in monocot, it is very distinct in dicot. Medullary rays are present in dicot, whereas they are not present in the monocot, and they are responsible for the secondary thickening in dicots. And cambium is of course present in the dicot, is not present in the monocot. Now if you look at their diagram you will understand in much